Sam, and I'm the president of 1 Million Solutions in Health. And we are thrilled today to be bringing you this webinar on the new gold standard in quality control of biospecimen studies. So thank you all for attending today, and we look forward to a very insightful and informative webinar. And I will just do a few introductions about ourselves and about the speakers, and then we'll get the presentation underway. So at 1 Million Solutions in Health, our goal is to improve healthcare and life sciences one solution at a time. And that is an example of what we're doing today, is we're bringing you a solution that you can implement in your area of life sciences and drug development. And we're very excited to be having this opportunity to share this great information with you. At 1 Million Solutions in Health, we help people connect around the globe, whether it's to be accelerating life sciences research, improving healthcare, or sharing patient and consumer-focused ideas and solutions. And we do that through various ways, whether it be through live events or webinar events like the one today, which is being recorded. We also offer discussion forums, articles, and we really hope to connect people. That's our true goal. And whether you're in academia, if you're in the government, you're an entrepreneur, you're a business, we want to work with you to bring new science to your organization. So let me give you a bit of a background today about the webinar. First of all, this webinar will present a one-of-a-kind technology that has the potential to revolutionize the industry. Experts from Metanomics Health, also from the Integrated Biobank of Luxembourg and the Medical University of Graz, will discuss the importance of human plasma quality control and a novel assay to improve it. With sample quality being the critical determinant in the success of clinical trials, it's more important than ever to invest in obtaining quality samples for biopharmaceutical research and biobanking. Sample quality is also critical in the development of biomarkers, companion diagnostics, and molecular diagnostics. So, in an effort to shed light on the importance of plasma quality control, Metanomics Health has teamed up with us to create a powerful webinar featuring these three prominent figures in the industry. These worldwide experts will discuss the overall issues with quality control, with a particular focus on human plasma EDTA plasma. So, let me tell you more about our terrific speakers for today. Our first speaker will be Dr. Kurt Zatlukal, and let me just tell you a bit more about his background. So, Dr. Kurt Zatlukal is professor of pathology at the Medical University of Graz, Austria, and is director of the Christian Doppler Laboratory for Biospecimen Research and Biobanking Technologies. His research focuses on molecular pathology of diseases, as well as biobanking and related technologies. He coordinated the preparatory phase of the European Biobanking and Biomolecular Research Infrastructure within the 7th EU Framework Program. Furthermore, he led in the FP7-funded large integrated project, SPEDIA, the development of new European standards and norms for pre-analytical processing of tissue samples for molecular testing. He is and has been involved in developing the ethical and legal framework for medical research and healthcare. He is a member of the Austrian, I'm not going to say this correctly, and the Scientific Board for Genetic Testing and Human Gene Therapy at the Austrian Ministry of Health, and also member of the Austrian Standards Institute. He was a member of the OECD Task Force on Biological Resource Centers and the Roadmap Working Group of the European Strategy Forum on Research Infrastructures. Moreover, he contributed to the OECD Best Practice Guidelines for Biological Resource Centers, the regulations for genetic testing of the Austrian Gene Technology Law, and was member of the Bioethics Commission at the Austrian Federal Chancellery. He has published 185 scientific papers and was co-inventor of 15 patent applications. Our second speaker will be Dr. Faye Betsu. Since February 2010, Dr. Betsu is Chief of Biospecimen at IBBL, where she is directing the activities of the biorefinery and the Biospecimen Research Laboratory. She is a molecular biologist with over 18 years of experience in molecular diagnostics, disease-oriented biobanking, and biospecimen research. Dr. Betsu holds 3 patents and is the author of 70 peer-reviewed publications, including many fundamental and experimental works on biospecimen research. Dr. Betsu's research background is in the molecular microbiology area, including molecular diagnostics, immunophysiopathology, and vaccines. She holds a biostatistics degree, a PhD, and an authorization for direction of research from Paris V University. She is also a coordinator of a university certificate on Principles of Biobanking for Clinical, Biological, and Environmental Biospecimens and Bioresources at the University of Luxembourg. Faye is a member of the ISBER Education and Training Committee and chair of the ISBER Biospecimen Science Working Group. And then our third speaker will be Dr. Philip Schatz. Dr. Philip Schatz is the head of biomarker program at Metanomics Health, a BASF group company, and since 2012. Dr. Schatz has an in-depth understanding of biomarker discovery and validation, assay development, and commercialization. He oversees the biomarker program, which provides metabolic biomarkers to support pharma-enabling and diagnostic applications in various indications, such as congestive heart failure, type 2 diabetes, neurodegenerative diseases, and multiple oncological applications. Prior to Metanomics Health, Philip worked for 12 years in the diagnostic industry. He holds a Master's of Science in Biotechnology Engineering from the Technical University Berlin and a PhD in epigenetics from the University of Saarland. So with no further ado, I will turn it over to our first speaker, and I will get presentation right there. Dr. Kurt Zatlokal will be speaking first, and just wanted to let everyone know that you can put any questions you have for the speakers into the chat box. The chat box is on the right-hand side of your screen, and what you just do is you address your questions to either the moderator at 1 Million Solutions in Health or to myself, Dawn Van Dam. And then I will ask the questions and the speakers will answer them later. We will also be putting up a survey to everyone at the end, so you can give us some feedback on this presentation and tell us what you'd like to see for future events. So with no further ado, I will turn it over to Dr. Zatlokal. Thank you.
Thank you for your introduction. In my presentation, actually, I want to highlight 2 topics. First of all, making the case why is standardization of sample pre-analytics so important? And furthermore, is what now are the specific pre-analytical requirements for metabolomic analysis, and I will highlight this with some examples from the research programs we have been involved in. The importance of reproducibility of scientific data has recently been highlighted in a cover story by The Economist. For instance, they found that many of landmark studies cannot be used by other laboratories. For instance, highlighted by a report from Amgen that they found that only 6 out of 53 landmark studies can be reproduced. So quality is an issue. And the question is how we can reduce the variations in scientific data. Big problem here is that even most advanced analytical technologies cannot produce better data than the quality of the biological material. So we have to take care the whole workflow from the patient, from the operations theater, from the clinics, to the analytical pipeline, that the biologically relevant imprint is maintained. And that's the same whether we talk about basic research, biomarker research and development, or drug development and target discovery. There is important knowledge developed in the environment of biobanks how to ensure the proper pre-analytical processing of samples for molecular analysis. These have been particularly addressed by 2 major programs, one under the umbrella of the National Cancer Institute in the United States, another one funded by the 7th Framework Program of the European Union, the SPEDIA program, and these 2 initiatives try to do systematic investigation how the various steps in the pre-analytical process impact on various analytes in biological specimens. The challenge that these programs are facing is that there are several steps from the patient to the analytical procedure, and these slides highlight some of the steps critical for the processing of tissues. Problem is that some of these steps can be well standardized, but others not, because they depend on the specific requirements of the patient. For instance, you cannot control duration of surgery, impacting on ischemia, medication given during surgery. So we all have to accept there will be some variation. This means when we cannot avoid variation, we have to know exactly what the impact on our analytical result is of these variable factors. And this becomes particularly important when we talk about the metabolome. On the one hand, the metabolome is so highly informative on the health or disease status of an individual because it integrates both environmental and genetic factors. But this at the same time makes the metabolome highly responsive to environmental factors and factors of the pre-analytical process. In order to investigate these parameters, we have performed studies during surgery in patients, this is liver surgery, in the context of a clinical trial protocol, where we could collect samples from the start of an operation and then could follow in the same patient how biospecimens respond during the various procedures and steps. In doing an metabolomics profiling, one can immediately recognize that a sample responds to ischemia by increasing lactate and influencing glutathione concentrations. Now, having a more comprehensive summary on these changes in the principal component analysis of several metabolite profiles, and these are data from different individuals. All these lines correspond to the sample collected at different time points from a given individual, that you see an immediate change in this metabolic profile already after 30 minutes. This means you have to very well standardize exactly the time point when you collect the sample. Otherwise, you can hardly compare results from samples having different ischemia types. The next question was, how does the lifestyle influence the metabolome? And to answer this, we have performed a study in healthy volunteers. And actually, we monitored the variations of the individual metabolome over a 10-day study. There were 7 days, a regular lifestyle, well documented on all key parameters, and we collected during the day 6 times saliva, all the urine, stool, and blood. And then there were 2 days of lifestyle with a highly standardized diet. On the last day, highly standardized diet and standardized physical activity. So we wanted to see how strong does lifestyle impact on the metabolome of an individual. And the next slide summarizes these results obtained by NMR spectroscopy in a 2-dimensional principal component analysis. And these different colors highlight the metabolic signature of different individuals. So what you can immediately recognize that each individual has its highly personalized metabolome, like the genome. But also you see there is some significant variation in the samples collected at different time points. Having a closer look into these variations, like here, the samples collected from one individual during the different days of the study, visualized in the different colors, you can see there is a big variation from day to day, and even standardized diet does not really avoid this variation. And of course, physical activity had a significant impact. So what's the conclusion we can draw from this observation is, if you cannot really standardize lifestyle, which is not easy in a healthcare setting, you have to take multiple measurements. Then you can normalize for this, and then you get highly specific metabolomic profiles for individuals. Next question was, how stable is a metabolome upon storage? And so we did a pretty demanding setup together with Fraunhofer, IBMT in Germany to simulate various storage conditions. And what we found is, by this simulation, that it's not so much the storage temperature, it's the retrieval process that has an impact. And we have simulated storage and retrieval is -80 freezers, storage in liquid nitrogen gas phase, liquid nitrogen liquid phase, and a system that has also a temperature-controlled retrieval environment. And we found that free proline is a very good surrogate marker to monitor changes. And you can see that even liquid nitrogen, there is a significant increase in free proline upon repeated storage and retrieval. And actually, we found only a modest variation by storage in liquid nitrogen gas phase. Again, what we can learn from this is it's not the storage temperature, it's the retrieval process that determines stability and robustness of metabolomic signatures in samples. At the end of my presentation, I want to acknowledge all the co-workers and colleagues that contributed to this data and work, and last but not least, the funders who enabled this work. It was funded by the European Framework Program, research program from the Austrian government. I thank you for your attention.
Thanks so much for your information and your insightful review of all the data. We really appreciate it. So we're going to turn next to our next speaker, and that will be Dr. Biospecimen, who will be talking and sharing her information with us, and we'll go from there. Thank you so much. Over to you, Dr. Biospecimen.
Thank you. Hello, everyone. Thank you for the introduction. So the title of this presentation is Quality Control of Fluid Biospecimens in Clinical Biobanking. And before starting, let me have a quick introduction on IBBL. As you know, Luxembourg is a tiny country, and IBBL is the National Biobank of Luxembourg. Of course, in a country of 500,000 citizens, you do not expect to have more than one biobank. So the vision of IBBL is to be an international center of excellence in biobanking and accelerate introduction of personalized healthcare for the benefit of the people in Luxembourg. We do collect biological fluids, tissue, and associated demographical, clinical, biological, pathological, and pre-analytical data. We also provide services in the perimeter of biobanking activities including the collection, processing, quality control, storage, and distribution of specimens. And IBBL is also at the center of a consortium in Luxembourg, which is called the Personalized Medicine Consortium. The focus of our collections is cancer, Parkinson's disease, diabetes, and there's also a healthy population collection. And the common thread or the common theme across all these collections is microbiome. So one of the reasons why IBBL is called integrated is because we integrate biospecimen research in the scope of method validation and the biobank's accreditation to ISO 17025 standards. The focus of accreditation is all the quality control assays that we perform on the samples in order to obtain objective evidence that requirements for a specific intended use are fulfilled. And this is the ISO definition of quality control. Now, accreditation means that every method has to be validated. And how is this done? First, a sample processing method, which is performed by the biobank, is validated through application of quality control assays on the processed samples. Second, sample quality control assays, which are performed either by the biobank or by an external laboratory, are validated through formal analytical method validation and proficiency testing, PT. Now, during validation of a sample processing method, pre-analytical variables must be examined and their potential impact should be evaluated. For biological fluid biospecimens, potentially critical in vivo pre-analytical variables include position of the patient during blood collection, fasting, smoking, stress conditions, circadian rhythms, menstrual cycle, pregnancy, physical exercise, as Professor Zapukal also mentioned. Again, for fluid biospecimens, potentially critical in vitro, this time in vitro pre-analytical variables include blood collection tubes, anticoagulants, clotting time, time of incubation before centrifugation, or storage conditions. I believe you are all knowledgeable about the ISBER, the International Society for Biological Environmental Repositories. And the ISBER Biospecimen Science Working Group has advanced the field of biospecimen science and biospecimen quality assurance and quality control in several ways. As an example, a standard pre-analytical code, or a SPREC, has been developed, and I will show you in the next slide what this is about. So a SPREC has been developed, as well as free tools facilitating this SPREC implementation in biobank databases. Also, potential new quality control tools have been identified and published by this working group. Apart from that, an ISBER-endorsed proficiency testing program has been developed by IBBL and is provided annually. And a fourth example, which is a very recent development, is an external quality assurance survey that has also been developed by the group and which is available on the ISBER website. Okay, now what is the SPREC, the standard pre-analytical code? We will look at one example on the top concerning fluid specimens. So the SPREC is, as the name says, it's a standard pre-analytical code of 7 letters, 7 elements, which are informative of the sample's pre-analytical history, or at least the most important pre-analytical variables of the specimen's history. As an example, if we have to do with a sample which is coded as U24PIX-BANAJ, we will know that this is about a 24-hour urine specimen, which has been collected with protease inhibitors. That's what the PIX means, which had less than 2 hours precentrifugation delay at 4 degrees. This is what the B means. A means that the sample had the first centrifugation at 3000 G room temperature without break. N means that there was no second centrifugation. A, that there was less than 1 hour post-centrifugation delay at 4 degrees, and J, that the long-term storage was in 5 ml polypropylene tubes at -80. There is also a SPREC for tissue specimens, and in general, this SPREC allows biobanks to report the pre-analytics of specimens, of course, when these pre-analytics are under control, when they are known. If the pre-analytics are not known, then the SPREC will include the letter or the code X. When the pre-analytics are not known, especially in historical collections, then quality control tools are needed. Quality control tools are needed, which are either predictive of the feasibility of a downstream analysis, or quality control assays, which would be diagnostic of the upstream pre-analytical history of the sample. Our ideal quality control biomarkers would be ubiquitous, measurable by a widely accessible method, and which show an on-off response to the specific pre-analytical variation. Examples of such quality control tools are the soluble CD40 ligand in serum, which gets completely undetectable after a certain time of post-centrifugation delay or of exposure of the serum to room temperature. However, as you can see on the left, this soluble CD40 ligand is not affected by freeze-thaw cycles. It is only affected by exposure of serum to room temperature. So this quality control tool, the soluble CD40 ligand, only applies to serum. It does not apply to plasma, and it only applies to post-centrifugation delay. It does not apply, it is not usable to assess pre-centrifugation delay. Another example of a quality control tool is the protein S activity in plasma, which again this time gets almost completely undetectable after a certain time of storage of the plasma at -80 degrees or even in liquid nitrogen. So other examples of quality control tools found and published by the ISPER working group include, so here we have transferrin receptor measured by ELISA, or potassium measured by potentiometry, which can help us assess serum pre-centrifugation delay. Now in this graph, to better understand, the blue and the red correspond to the minimum and the maximum of the reference range at the baseline, or the minimum and the maximum of the analyte after a certain pre-centrifugation delay. The reason why this example of transferrin receptor is a good example of quality control tool is because, as you can see, the minimum level after 8 hours of pre-centrifugation delay is significantly higher than the maximum level at baseline. Right. Now, other examples, ascorbic acid measured by chromatography could be used to assess serum or plasma pre-centrifugation delay, but also suboptimal storage conditions, -20 degrees instead of -80. Now, for plasma storage conditions, dopamine or vitamin E measured by HPLC could be used to assess plasma suboptimal storage conditions. Things are more difficult where it comes to urine. Dopamine could also be used to assess suboptimal storage conditions for urine, for dopamine. However, the example of alpha-1 antitrypsin is a more tricky example or possibility of a quality control tool. And why is it more tricky? The reason is, as you can see, because the baseline reference range of this analyte is huge. So it is very difficult, if not impossible, to use a quality biomarker as a quality control tool when the reference range is so big. So in ISPER, we are working towards the goal of establishing a panel of quality attributes or quality control tools where each quality attribute will allow us to assess specific pre-analytical conditions for each specific fluid biospecimen type. Another example, for cerebrospinal fluid, cystatin C would help us assess again the specific pre-analytical variable of -20 degree storage instead of -80. Finally, we at IBBL, as I said at the beginning, we are introducing each year new schemes in the annual proficiency testing program. This proficiency testing program allows each participant to assess the accuracy of his quality control testing relative to the performance and the measurements made by participants all over the world. Current schemes in this proficiency testing program include schemes for DNA quantification and purity, RNA integrity, cell viability, tissue histology, but also there are schemes for DNA extraction from whole blood, RNA extraction from whole blood, and DNA extraction from FFPE. Now, and this is, I will end my presentation here, certain quality attributes, quality control tests may be developed and validated, of course, by external suppliers, by external specialists. And in this respect, this last scheme, plasma metabolomics quality assessment, is a scheme which is performed in collaboration with Metanomics. And let me make at this point the transition to the next speaker who will tell us more about this plasma metabolomics quality assessment by reminding you what I previously said about the soluble CD40 ligand, that this is a quality control tool which allows us to assess pre-analytics only for serum, not for plasma, and only for pre-centrifugation and relatively long, post-centrifugation delays. So some things we were missing, some quality control tools missing, and the next presentation will bring some answers to these gaps. Thank you.
Great. Well, thank you so much, Dr. Betsu. We really appreciate your presentation. And I think this is all building nicely and going to be a great opportunity for people to ask tons of questions. So just a reminder, if you do have any questions, to put them into the chat box, and then we'll get to those after the end of the 3 presentations. So next, we're going to turn to Dr. Philip Schatz, who's going to talk about the new launch of this gold standard in quality control biospecimen studies. Over to you, Dr. Schatz.
Thanks a lot for this nice introduction, and thanks as well to Dr. Betsu and Dr. Zatoka for those brilliant presentations which highlighted the importance of high quality control and the impact of very important clinical features such as dopamine and several vitamins which we have just seen. Now I'd like to introduce you to a solution to this pressing issue of bad quality samples in biomarker research, and this is provided by a product which we have launched at spring this year in London at the Biobank Global Congress and in Orlando at the ISPO annual meeting in Florida, and it's called MXP Quality Control Plasma. And this helps to upgrade existing quality assurance to a phase in which we can truly quality control the sample, not relying on documentation, but really understanding what the impact of the individual in the individual sample is to the overall quality of the specimen. I'd like to start with the first example of our internal research, and here we have analyzed samples derived from a clinical trial which used ongoing sampling of at the very same clinical site over a longer period of time. And what we've done is that we have looked at a certain ratio of 2 different quality measures, and we've looked at that over the time. And what you can see is that at a certain point in time, we suddenly observe a subgroup which suffered from insufficient quality, and this was true for both individuals suffering from a disease and those which do not suffer from disease, meaning that this was an independent measure, independent of the overall health status of the sample, however, highlighting the importance of continuously quality control, enabling additional training at that site, and potentially excluding those samples from downstream analysis. We do the assessment of the quality of the sample in 3 different measures. So first of all, we analyze the used plasma or blood collection tube in the first place, being able to distinguish between EDTA plasma, heparin plasma serum. Second, we assess the time prior centrifugation, so whether or not the time between blood draw and separation of the blood cells has been delayed. And at the third place, we analyze as well the storage of the plasma sample itself and whether the separation of the blood cells has been done according to standard operating procedures. This all translates at the end into one single quality score, which provides you with a comprehensive overview of the quality of a given individual sample. We have beta tested this protocol together with a company which most of you should know, that which is Bayer Pharma, and they donated us or provided us with sample material blinded to us suffering from different confounding factors. In the first place, they selected different anticoagulant compounds such as EDTA, citrate, and heparin, and with just 3 different analytes, we were able to clearly distinguish each of those individual coagulant groups and were able to identify those samples which truly used EDTA plasma blood collection tubes in the first place from all others. In addition to that, we have as well introduced other confounding factors such as the delay of the blood prior centrifugation and the longer incubation of plasma material after separation of the blood cells. And last but not least as well, the introduction of additional freeze and thaw cycles to the plasma samples. What we've seen is that our test was capable of identifying correctly 80 out of a total of 20 samples not suffering from any confounding factors as truly being of high quality, translating to a specificity of 90%. And by analyzing confounded samples, we were able to identify 98 out of 100 samples, translating into a sensitivity of the same assay of 98%. The assay works very easy. The starting material is, and this is true for all assessments which I've already introduced you, so everything is based on a single plasma sample, which is then further processed is very easy chemically, including an extraction cleanup step and a derivatization step in order to be able to analyze the samples with GCMS. The analytes are afterwards analyzed with the algorithm integrated into a software which is provided by us into a data or sample quality report, which allows the assessment of all those different aspects which we have already learned to. Such a protocol looks as it is highlighted on the right side of the slide. It's composed in a way that at the top of the report we see an overview about the entity of all samples analyzed in one batch, and below that we have individual assessments of different samples, providing each and every time 3 different informations: the identity of the used blood collection tube, the assessment of the blood processing control, and the assessment of the plasma processing control. We use a traffic light system to color code the quality of the sample, and this is provided for each different assessment individually, which can be explained as follows. The blood processing control, if it's highlighted as a green sample, the blood cells have been separated within 2 hours after initial blood draw. If it's medium quality, it's between 2 and 6 hours, and if it's above 6 hours, then the sample is highlighted as a red sample, meaning that it suffers from insufficient quality of the blood processing control. A similar system is provided for the plasma processing control, where we as well see that elongated time leads to a lower score, translating into a red color coding of the individual sample. However, this color coding provides you only with an overview which you can quickly assess. However, a more accurate scoring is given by individual numbers for each and every of those assessments, and as well translating into an overall quality score which provides you with a comprehensive scoring of the entire sample. I'd like to share with you now a second example from our internal research, and here we started with analyzing with the discovery of a new biomarker which should be able to distinguish between cases and controls. However, in the first place, we see that we observed an insignificant p-value, meaning that the two groups were not able to separate from each other. In a subsequent quality assessment of the very same samples, we were able to identify a group, which is here highlighted in green, which had a very good quality, and a second group, which is highlighted in red, which suffer from low quality of the used sample material. Due to the information we got from our quality assessment, we were able to identify that those samples suffered from oxidative stress. And by excluding those samples from the data analysis, suddenly the difference between the two groups became apparent, and we generated a significant p-value. In other words, a study which would have been a deal breaker, so enter stop of the discovery process, suddenly turned into a big success, and we were able to identify a new biomarker for that particular disease. The service we are here offering is actually available in two different manners. So we can either get samples from your facilities and analyze the samples in our laboratory and provide you with a report. Or alternatively, we can train your staff in order to be able to run those analyses in your own laboratory, which of course would require a GCMS device. The price range ranges from 5K for a low amount of samples up to approximately 100 or below 100 euros per assessment per sample if it's performed in a service manner. For customers who are more interested in high throughput analysis and have a higher demand of quality assessments, we offer as well the licensing model, which comes with a much lower price depending on the amount of samples you analyze. The system here works very similar to a mobile phone contract. We provide you with the software and you purchase keys which can then, or which allow for a certain amount of analysis which comes with a certain price tag depending on the overall amount of samples you analyze. I'd like to finalize my talk with the different areas of application in which we believe this new service is valuable. So starting with the pharma and diagnostic industry, here we really provide a tool enabling the upgrading of the existing quality management towards a quality control, knowing what you're doing, and as well the monitoring of clinical trials ongoing in order to verify that the collection has been done correctly or if additional training is required. In addition to that, prior purchasing high valuable sample material, one could assess such a collection in order to justify investment. From a biobank perspective, ongoing monitoring is as well very valuable and can be used to certify that the internal protocols are very accurate and very reliable, and the derived quality of your samples are worth the price you're offering to your customer. In addition to that, obviously you have a tool in hand to train your own staff. Last but not least, from a clinical research organization point of view, again, you have here a tool in hand which enables you to prove that you are offering superior quality in comparison to your competitor, and enabling this not only in a single point, but ongoing in as a monitoring tool in ongoing clinical trials. With that, I'd like to close and thank all the participants, and we are all three now ready to answer your questions.
Great. Well, thank you so much, Dr. Philip Schatz. I'm very excited to start to answer the questions and understand how we can address what everyone would like to learn about further. So I'm going to start back with Dr. Kurt Zabucal. The first question, I think, is geared towards you based upon when it came in. Let me just get to it here. Okay, how is the experience from the slides and the information that you presented? How are those presented studies implemented?
Yeah, so most of this data were generated in the context of the SPEDIA project, which was funded by the Framework Program 7, and the call for this program requested to provide the scientific data that leads to new European standards and norms to improve reliability of molecular diagnostics. And there was the committee, the European Committee for Standardization, was partner of this program, and these data are now used by this committee to develop new European standards and norms for diagnostic purposes, and they are aligned with the current standard 15189 for diagnostic laboratories.
Great, thank you very much. So again, if other people would like to ask questions, just put them into the chat box. That would be fabulous. So the next question is for Dr. Faye Betsu, and the question is, what are the unanswered questions in the area of QC for clinical fluids?
Right. Can you hear me? Oops.
Yes, we can hear you fine. I was just muted, so yes, we can hear you fine. Yeah.
Yeah. So actually, there are many unanswered questions, and some were mentioned throughout the talks. For example, if and what is the impact of freeze-thaw cycles on clinical fluids, serum, plasma, urine, cerebrospinal fluid? We don't know. For specific types of biological fluids, actually we know almost nothing on the pre-analytics. This is the case for urine, cerebrospinal fluid, saliva, which is a very complex fluid biospecimen. So all questions are unanswered concerning the pre-analytics in the scope of proteomic or metabolomic analysis. Another example of unanswered question, the impact of pre-analytics of whole blood in the scope of cellular functional assays. So we have a lot to answer.
Okay, great. Well, thank you very much for that answer. And I think there's a follow-up question here, which I'll just ask you again next before we move on. How does the ISBER Biospecimen Science Working Group plan to address some of these unanswered questions?
Well, as I said, through international collaboration and inter-laboratory validation. More specifically today, collections are being constituted in the United States, in Europe, in Australia by members of the ISBER working group, and these will be used for quality control validation. So it's an ongoing effort, and it's an effort which is being done without specific funding, but we are lucky to have a group of very motivated biobank scientists who dedicate time and efforts towards this important goal. And if anyone in the audience wishes to join this effort, you can either contact me or volunteer in ISBER.
Great, excellent, thank you. So I have a question here for Dr. Philip Schatz, and I think you've answered most of it, but I always like to repeat the question just in case you have not. This one probably came in before you answered it. But the question is, has Dr. Schatz's data been published? And also, what are the costs for the analyses?
Everybody able to hear me now? Yes, okay, perfect. So this has been published in Clinical Chemistry in the spring this year. The first author is Dr. Beate Kamblage, can be found online. It is open access. The second question regarding the price tag, we have kind of a more complex price situation. So we have two options, service option and the licensing model. and the price depends on the amount of samples you're sending over. I would ask the audience if there's more questions in that respect to simply contact our business development department in order to get all the detailed information about the price test, or simply refer to the presentation I've just provided or shared.
Okay, thank you. And maybe what we'll do is we'll just provide the business development email address or something like that, either in a follow-up email to this, or if we can stick it into the chat box, we could do that if we get it over the next few minutes. So another question that we have here is perhaps we could have everyone answer this question. Why do I need quality control if I strictly follow my SOPs? So I'll just open it up to any one of you to answer, and then the other two can follow. Or should I just choose someone? Go ahead, Faye. You go first.
Well,
go ahead.
Yes, it's a good question. Why do I need quality control if I strictly follow my SOPs? First of all, SOPs are never followed 100%. There are always what we call non-conformities, and this is sometimes outside our control because we work with hospitals and clinical centers. We do not have control over everything, and non-conformities are part of the everyday work. So 100% compliance does not exist. Okay. Then, because, as I said, we collaborate most of the time, most of clinical biobanks, but also population biobanks, we collaborate with specimen collection centers. So when they are located in the hospital, we depend on medical, paramedical staff, clinical research nurses, over whom, most of the time, we do not have control. So the SOP may specify that the timing between collection and centrifugation must not exceed 2 hours, and the clinical research nurse may document on the sample collection form the timing of collection. We document, of course, the timing of reception of the sample at the biobank. But this is not objective evidence. Okay, so this is one part of the answer. And the second part of the answer is if we need to supply samples which are accompanied by a certificate, then the only way to do this, to provide certified samples, is via a quality control.
And thanks.
Okay, thank you.
May I add one aspect to that? So I would even phrase it a little more dramatic, meaning that the difference is actually among trusting in quality, which is documented, of course, and the compliance is documented, and knowing. So while controlling the quality in the individual sample which you actually analyze, then it's not just trusting anymore, but you know the answer, you know whether the sample is correct. In addition to that, typically quite some aspects of the overall logistics of a sample is not 100% documented. So for instance, transportation and transfer even within the very same institute, not in each and every case is 100% documented. And the only way in order to have a comprehensive overview is really checking the individual sample prior to the important analysis.
And also, when it comes to historical collections, today the majority, the millions of samples which are resting in biorepositories belong to historical collections. Because today, right, we are aware, we are sensible to the importance of pre-analytics, we have SOPs, we document things, but don't forget the word SOP in the biobanking community appeared 8 years ago. So all the other collections, historical collections, have no accompanying pre-analytical data whatsoever. So quality control is the only way there.
And another aspect, I mean, we are all doing research here, and in some cases, during the time when a sample has been collected, you simply didn't know that certain aspects could have an impact on the quality of a certain analyte you're interested in. And then, for instance, time between blood draw and centrifugation was not documented. However, it later on turns out that this has a huge impact on your individual analyte, and then you would like to assess it.
Okay, thanks. Kurt, did you have anything you wanted to add to that, or did Faye and Philip cover everything?
Perhaps another aspect we should keep in mind is that I see a lot of potential of metabolomics also in diagnostic applications. And then there is the problem that you have limited opportunities, high level of standardization in a routine healthcare setting. So I think there is increasing need also for quality control parameters, particularly if you want to apply metabolomics in a routine healthcare setting.
Actually, you're a great setup person to someone else's question there, Kurt. So one of the questions that's come in is, having seen the strong impact of pre-analytical factors on metabolites, the question arises, how can this be implemented in a routine healthcare workflow? So do you have any thoughts on that?
Yes, at least there is some data emerging showing that it's not all metabolites that are so responsive to pre-analytical factors. Actually, some metabolites are pretty robust, because otherwise we couldn't measure glucose and lipids in a routine healthcare setting. But others are highly responsive. And at the moment, we are still lacking the knowledge to know in advance which metabolites are highly responsive and which are more robust. So, and I think at least as long as this knowledge is not really established, we have to be very careful about these pre-analytical parameters. So it really depends on the question you want to analyze, and particularly which metabolites you're interested in, how strongly you have to control your pre-analytical process in order to guarantee reliability of data.
Okay, any other comments from Faye or Philip?
Yeah, I'd like to add something. Since we are pretty much doing what Kurt just described here, so we are running here our own clinical metabolomics program in order to discover new biomarkers for certain indications and diseases. And this is exactly what we're doing. So we've not only look what is the difference among cases and controls, but we as well look which metabolite, which analyte is more impacted by or influenced by certain confounding factors, and we control for that in a way that we don't select those biomarkers. So at the end, we use only those biomarkers which are robust against confounding factors, and that's, by the way, not only pre-analytical phase, but it's as well age, gender, BMI, a lot of other factors where we kind of pre-select only those markers which are stable against them in order to make them fit for purpose for clinical use.
And Faye, anything else that you wanted to add?
When we talk about implementation of quality control assays in a routine clinical setting, personally, I do not think we are thinking at implementing routine quality control on all the samples coming through a routine clinical setting. What we are talking about is implementing quality control on the outgoing samples, so upon distribution for specific research applications. That is the point in the workflow where these quality controls would be applied. So not on all incoming specimens from the routine clinical setting, but on outgoing specimens for specific applications.
Okay, any other further comments before I move on? Okay, so someone has asked, where can I find the SOPs for correctly collecting and storing all biospecimens? And perhaps if there's a website link, we could just stick it in the chat box.
Yeah, I might give it a start. So in principle, we have collection protocols for quite a variety of different specimens, both from animals and tissue and cell culture. However, this is only available under CDA, but we are at the moment working together with Professor Satlokar and other co-workers like Dr. Fabitsu as well on in pre-competitive alliances under the umbrella of the BBMRI, and the idea of those corporations is, among others, as well the establishment of guidance how metabolomics can be best practiced, and sampling obviously is a big part of that. But I'm sure that Dr. Zatlokar, Dr. Vetsu would like to add to that.
Yes, perhaps a brief comment from my side is at the moment we have not, at least I'm not aware of, internationally harmonized SOPs, because SOPs are so specific for a laboratory, otherwise it's not an SOP. But we need this guidance, how SOPs should be structured and which parameters should be defined in an SOP. And I think there will be a big change in our landscape, maybe somewhere in the mid of next year, when the Committee of European Norms will publish the technical specifications, which give very good guidance on how to structure SOPs and how to at least document or reduce pre-analytical variables.
Maybe the question about correct SOPs is, of course, it's a big question, but the answer maybe is, we cannot have such an answer, say, what is the correct SOP, because there is no such thing as correct SOP. The question should be, what is the correct or acceptable SOP for a specific downstream application? So there are some elements in an SOP that should be respected for a specific downstream application. For example, for metabolomics, the delay should not exceed 2 hours, and the temperature should not be room temperature. The specimen should be put immediately on ice. There are some things, such examples, but we will. One thing is that the most important is to document, because there will be no time where all the laboratories in the world will apply the same SOP. This is obvious, and this should not also be the case, because then we lock ourselves in a system which precludes us from certain discoveries. So the most important is to document, keep records of what exactly has been done, how specimens have been collected, processed, and so on. There are several references, journals, on websites. CTRNet has published SOPs in Canada. There are a series of SOPs in biopreservation, biobanking. There are protocols all around. Okay. Again, to say that this is the correct protocol, this is the protocol that one should apply, there is not such things. Yes, the respect for certain parameters, for certain downstream applications, and for the rest, documentation.
Great, thank you so much. Okay, just want to recognize to everyone that we do have a number of more questions, and we will take additional ones, but since it is a little bit past the top of the hour, if you do have to leave, please just fill out the survey before you go, and also if you want to ask a question and you have to leave, just send the question in anyway, because we'll continue to answer them for the next few moments. So another question for our panelists is, did I get Dr. Schatz correctly? The MXP system can be used for plasma only, not to test serum. Is that correct or not?
This is absolutely correct. At the moment, we have only the plasma product launch. However, additional test systems are currently under development or in the development.
Okay, and while we have you on the line, is the service, another question for you is the service model performed at all of your sites?
At the moment, we offer the service only from our Berlin facilities.
Okay, great, thank you. So again, just a reminder to anyone to send in any other questions. And I am going to go to my next ones that I have here. Why are pre-analytical effects different for blood storage and plasma storage? So again, opening it up to anyone.
Why are pre-analytical effects different for blood and for plasma storage? The answer is that when it comes to blood storage, the pre-analytical effects are mainly due to the impact of the cellular elements which are present in the whole blood. Whereas for plasma storage, we have gotten rid of most of the cellular elements. So effects that are happening in the plasma have to do more with the protease activities, remaining enzymatic activities in the plasma, but not with, for example, release of components from cells, cell lysis, cell activation, so
on. Great. Anyone else want to further answer that question?
No, that's well covered.
Okay. Great. And also, we did get a question as to whether or not the slides will be made available. We will get back to people on that. We'll definitely be making available the Metanomics Health slides, and we'll see how we will do the other ones, if at all. But the webinar recording will be made available to everyone who participated and then up on our website additionally afterwards. So another question for the panelists is, what is the minimal size of the assay that Metanomics Health can accept? Can small sample sets, for instance, N of 12, be analyzed for reasonable cost to test this service?
I guess I should take that question. The answer is, in principle, we can go down to a single sample. However, this comes with a certain price. So we have a certain price limitation, which is 5K euro. However, there's at the moment, this has been introduced by Dr. Betsu, at the moment proficiency testing ongoing, and this has a different price structure, and in that way, at least at the moment, that is a way to kind of assess the service with a lower price tag.
So in the context of the proficiency testing program, the batch of samples to be sent is 4.
Okay, great, thank you. Anything further on that one? Okay, so why does the quality control work for EDTA plasma only? Are there more sample types than EDTA plasma only in the pipeline? And I think you may have answered a bit of this already.
Yeah, I've answered that. So at the moment, we are as well working on heparin, plasma, and serum. But there's more to come, but we have to start somewhere.
Great. Okay, thank you. What is the recommended volume and what is the minimum volume for the test?
We actually use 70 microliters for the analysis. However, we recommend to provide 100 microliter in order to have pipetting access and potential to duplicate or repeat the measurement.
Okay. Thank you. And so I'll just give one last call for questions because I have one more question to ask and I just wanted to see if anyone else has anything. And then after we answer this last question, I'll just ask each of our speakers to make some perhaps quick closing remarks. So the last question I have, unless more come in the next minute, is what is the biochemical or physiological background of pre-analytical effects on the plasma metabolome?
Okay. First, or part of the answer, I think, is lots has to do with anaerobic conditions. The main driver, and Philip, you may be more knowledgeable than me on this part, but I think the main driver is anaerobic conditions once, and the same applies obviously to tissue. I mean, as soon as the biospecimen is taken out of the body, be it blood or tissue, hypoxic conditions start, and metabolism is impacted by those.
Yeah, I would actually split the situation in two. There's either a biological effect or a chemical effect. The moment you have separated the cells and store your sample at frozen conditions, obviously only chemical effects take place, so basically oxidation. When the blood cells are still present, there's a lot of platelet activation and a lot of enzymatic activities ongoing derived from the cellular fraction. However, and Faye very nicely explained this previously, that in the plasma there's as well some enzyme activities left over, and this is even or true in a situation where you have anticoagulants added to the plasma, however, even more prominent in heparin or even serum, where there's no anticoagulants added, and this will change the levels of several analytes, and that's not only metabolites, but really a lot of other clinical important factors as well.
Okay, fabulous. Thank you very much. So I haven't received any additional questions, which is terrific because we've had tons and tons of questions. So I'll turn it over to each of you to give some closing remarks. And perhaps, Kurt, do you want to start us off?
I think it's very important raising awareness on the pre-analytical factors and quality of samples. And particularly, I see a lot of future in metabolomics, and not only in the research setting, but also in the routine diagnostic setting. Thank you.
Great, thank you. Over to you, Faye.
Just to underline again the importance of pre-analytics, biospecimen science, quality control for the improvement of resolution and robustness of future biomarker discovery and the reliability of all those research results. And I think really that also that because we're talking about metabolites may offer the most sensitive of these tools.
And over to you, Philip.
Okay, I'd only like to highlight that we all know that we all spend very much resources in accurate biomarker research, and as well in order to improve healthcare, and as well in biobanking. And I guess it became obvious that this is only valuable the moment we work accurately and when we enable ourselves and provide tools to assess that. And I guess it became clear to everybody that we have now a solution to that, and this hopefully will have a strong impact, and maybe in the future even on clinical routine. However, at the moment, we rather see this as a research tool.
Great. Well, thank you very much. I want to again thank Dr. Kurt Zatlakal, Dr. Faye Betsu, and Dr. Philip Schatz for their fabulous presentation today on this breakthrough technology. I think it'll be a game changer in the industry, and we really appreciate everyone's feedback and participation today and look forward to continuing the dialogue. So thanks again to all of you, and particularly to our attendees. Thanks, everyone, for participating today.